Hello, I'm back. So now we're going to talk about um, second part of chapter 1, the discoveries that were super important and essential for what we know today in microbiology. So everything starts with Robert Hooke in 1669. Look how old this is, super old. And these scientists described the cells that were part of how the organisms were made. So he called them little boxes, and then later they were called cells. So the first microbes were observed a little even before that, and they were called animalcules. Okay, so this is the microscope that they used to observe the first microbes. Even before that, because microbes were not something that was clear, they didn't really know microbes existed, there was a theory that whatever being that was being formed was like a vital force, and that vital force would come from matter and create an organism. So a piece of the wood on your desk could create a microbe, and that was what it was called the spontaneous generation theory. So according to this hypothesis, if you had something sterile, which means free of microbes, in a flask, and you just let it sit there, closed, hermetically closed, some life could be originated from that matter by what they call the vital force. Okay? And then some scientists were not very comfortable with this, including Louis Pasteur, that is one of the biggest names in microbiology. So in 1861, this scientist demonstrated that spontaneous generation was not really so cool as everybody else was talking about, and it wasn't necessarily true. So he wanted to prove that a cell needs to come from another cell. A cell cannot come from dead matter. Okay? So what was his experiment? So he did two important experiments to prove this. One of them was he had a nutrient broth in a flask, not sealed. And then the other flask that he had with the same nutrient broth was also heated, so they were both heated, but this one was sealed immediately. And then the results that he observed were that the one that was not sealed, he could see microbes growing. And then they learned that whatever growth that was showing there, when they looked at the microscope, it looked like microbes, right? While the ones that were in the flask that was immediately sealed did not have microbe growth. So where was the microbe coming from? The air, right? And then not everybody was really convinced that spontaneous generation was not the thing, and maybe the thing was biogenesis. Living cells arise from only pre-existing living cells. So to be able to prove this to to a bigger audience, he decided to use this experiment here. So what he did was he had a flask with a very long neck. He added a broth. He heated this flask neck to be S-shaped. You see how it's like the shape of an S? And he heated the broth inside that tube, okay, the flask. When he did that, no microbes grew because probably this S shape here would block microbes from the air to get into the bottle, okay? But when he got that flask and he tipped it and put it back in shape after heating, meaning whatever was here, the broth that was here, would be able to travel all the way here and have some contact with the air. And what was in the air? Right here. Microbes. right? So when he tipped the flask and put back in place after heating, he saw microbial growth. So that was another way to show that microbes were coming from the air. Okay? So this was an essential experiment to prove or to disprove spontaneous generation and to reinforce or support the theory of biogenesis. Okay? And then other experiments that Pasteur did were super important. So his work is related to discovered including relationship between microbe and disease, immunity, antimicrobial drugs. He also studied fermentation. So when we learn about fermentation, you're going to see how fermentation is a very amazing anaerobic process that you start with sugar and the bacteria can transform that sugar into other compounds in the absence of air, so anaerobic. You're going to see how amazing that is. Okay, and we wouldn't have bread and beer and wine and soy sauce and kimchi and other foods and other things that are produced by the industry if it wasn't for fermentation. Okay. He also studied food spoilage, which how can food spoil so quickly? Or some food spoil less quickly. It's all related to how much microbes can grow on that food and use that food as their source of energy and grow, right? So all these were experiments that were proved and demonstrated by Pasteur and were part of the golden age of microbiology. Okay? Look at him. That's the guy. So there are other scientists that did really important experiments in proving that microbes cause disease. There is another scientist, Robert Koch, that we're going to talk later, that has a very interesting theory about how can you prove that a microbe causes a specific disease. So we're going to talk about this in a following chapter. And that's pretty much what I wanted to tell you here. And I'm going to stop. So we have 3 chapters to cover in this first week, right? 1, 4, and 5. So I want to make it quick so we can focus also on the other chapters and cover all this content for you guys to be sharp and understand and grasp the content of these chapters. Okay, so I'll talk to you again in when we're describing what's going on in chapter 4. Bye-bye.